Science/biology teacher, Mr. Poser. Today we are getting into our second topic of Unit 6 on gene expression, which is how proteins are made. In our last topic, we discussed the DNA structure, the double helix structure, the double polynucleotide strands, meaning it's a really long collection of A's, G's, C's, and T's, which are nucleotides that are assembled to form like a twisted ladder or a double helix. And that's going to come back in a big way. Oh, and we also talked about how DNA copies itself through DNA replication, which we'll come back to in just a bit. But this is going to be important stuff for how we discuss how proteins are made, because in our last video, what we also talked about was the central dogma of biology, or the process by which a DNA is used to first make RNA, and then RNA is used to make proteins. And that's what this is about today. How DNA is converted into RNA, and then RNA is used to make proteins. All right, so the anchoring question here is, how is DNA like a language, and how is a protein like a sentence, right? So we got two different systems of lettering over here. Well, language, right? So how is DNA like a language, and how is protein like a sentence, right? So how do those two things tie together? We'll come back to that at the end of the video. And one of the biggest questions of all of biology really is, how does a molecule, DNA, determine your traits, determine who you are? That's what we're going to get into in this video. So how proteins are made? Well, it's through protein synthesis, which is the flow of information from DNA to proteins, right? So this is our central dogma. This is the same picture that we looked at in our last video. DNA is first transcribed into this single-stranded molecule called RNA. Okay, so here we are in the nucleus. The DNA is stored in the nucleus. It's transcribed into a messenger molecule called messenger RNA, or an RNA molecule. And then RNA is then translated into protein. So the message that this carries is translated, kind of like a language, into proteins, okay? So what we're really looking at here, what this process is called, in short, is called gene expression, right? And so what is a gene? It's a segment or a piece of DNA that provides the instructions for a cell to make a specific protein, right? So if a cell's genome, or all the DNA within a cell, is like an instruction book, a gene is the instructions to make one particular thing. One particular thing, okay? So a gene is used to make one specific protein. So if you can think of any protein, say like insulin or actin or myosin, each one of those proteins has one specific segment of DNA that the cell uses to make that protein. And that's it, okay? So for every protein, for every type of protein, there is a gene in order to help the cell make that protein. Okay? And if you don't recall from earlier this year, proteins have a wide variety of functions and do a lot for cells. Okay, I think we just put that proteins do pretty much everything for a cell, and that's true. Okay, now the types of proteins a cell produces depends on, well, its differentiation, right? And if in our last unit we talked about cell differentiation, this stem cell here, which is undifferentiated, doesn't have a job yet, is going to eventually become one of these other types of cells that's highly specialized. It's only good at one particular job, right? And what its job is depends on the genes it expresses, meaning that it depends on which segments of DNA it's going to read and make proteins for, right? That help make its job. For an example, muscle cells are going to make a lot of proteins called actin and myosin, those are what we call myofilament molecules that help it contract. Okay? It's not going to be producing digestive enzymes like an epithelial cell. So these two different types of cells are they all have the same instruction book, but they're just going to be reading different parts of that instruction book, and that's where it's cell differentiation. That's how it ties into gene expression. Okay, so gene expression really determines what a cell does. Okay, so this whole process, gene expression or protein synthesis, begins with a process called transcription. It's the process of copying a sequence of DNA to produce a complementary strand of RNA. Now, we're not copying DNA, right? We're not replicating it. Okay, that was last topic. We're not making a new copy of DNA. What we are making is a little segment, a little transcript of a gene. Okay, and we're making that transcript, that little copy of that gene, only that gene, out of RNA, and we're not making more DNA from it. Okay, so we're copying a sequence of DNA to produce complementary strand of RNA, and this occurs in the nucleus, right? So here we are in this whole process of gene expression. Here's our DNA. It gets transcribed into mRNA through the process called transcription. And the molecules that are going to be doing the transcription are called RNA polymerases. Don't get that confused with DNA polymerase, right? RNA is we're going to be making, well, RNA from it, messenger RNA. So what it's going to do, it's going to open up this DNA strand here, And kind of similar to replication, it's going to open it up and then make a new complementary strand, but it's not DNA this time, it's RNA. And it'll only make one strand of that RNA based off of one of the old two DNA strands. Okay, and one other thing about RNA is that instead of a T nucleotide, it has a U nucleotide. Okay, so U stands for uracil. So you're never going to see a T, a thymine, in a RNA molecule. All right, so let's just practice already. Can we transcribe this DNA strand? It's going to be a lot like matching DNA strands like what we did in our last video, in our last topic, but there's a little catch here. Okay, so we have one strand of DNA. What's the RNA strand, the transcript going to look like from it? Okay, it's going to be A goes with T as always, C goes with G as always, but, okay, check it out, A goes with U. Okay, so if you see a T in the DNA strand, it's going to go with A, but if you see an A in the DNA strand, it's going to match with U in the RNA strand. So it goes AGU, CCA, UGC, GAA. A goes with U. Apple goes to the umbrella or something. If you can think of something better than that, let me know. All right. Okay, so let's get to the next part of it. That's transcription. mRNA then leaves the nucleus and goes to a special structure within the cell called a ribosome. I don't know if we got quite into these in our unit 2 or in our, what would that be, our third unit this year. But in a ribosome, it's a structure in the cytoplasm. It's not technically an organelle. It's a structure in the cytoplasm or the endomembrane system where proteins are translated. And a ribosome is known as a protein factory of a cell. All right, so here we are in our gene expression. mRNA leaves the nucleus, okay, and what it's going to do, it's going to head down here to a ribosome. Okay, it's going to kind of be fed through this ribosome, and it's going to carry that message, carry that copy of those instructions, and it's going to, and the ribosome is going to use those instructions to build a protein. And we're going to see how that works here in just a second. So here's the nucleus. This is where our DNA is, and this is where transcription occurs. So mRNA has to exit the nucleus and head out into the cytoplasm and then find one of these ribosomes in order to translate and produce that protein. Okay, so the ribosomes are going to, well, on a bigger scale, they look something like this. The ribosomes are going to be the sites of what we call translation, decoding mRNA in order to produce a protein. And every single cell, every single living thing that's ever existed does this transcription and translation here. Okay, so mRNA is read 3 nucleotides at a time while passing through a ribosome. Okay, so check this out. Here's our long, stringy mRNA. It's our transcript of a particular gene, that set of instructions to make a protein. Okay, and now it's taking, this ribosome is taking in those instructions and building a protein. You see this long chain of these blue molecules here? Okay, it's taking these instructions and using them to put specific amino acids in order to make a protein, and that's what translation is all about. It takes, it reads a message 3 nucleotides at a time, so like AGU, CAC. It's going to be reading those 3 at a time in order to assemble an amino acid chain, and that's what a protein is. Okay, a protein is, way back to Unit 1, a protein is a very, very long chain of amino acids, right? And in order for that protein to work properly, it needs to have the correct amino acid sequence. And that's what translation is all about. The words of the language of mRNA are what we call codons, okay, which are 3 nucleotide mRNA sequences that code for amino acids. Okay, so check this out. What we just did before, we took a transcript of mRNA from DNA, right? And we got 3, I think there was like 4 sets of 3 letters, right? Each one of those 3 letters is called a codon, and one codon gets translated into one amino acid in particular. And how do we figure this out? Well, we use this chart over here, and I actually love this chart. Check it out. It's my mouse pad for my work computer over here. And it's not about guac, okay? These are your mRNA nucleotides, right? So what we're going to be doing here is translating that mRNA message into amino acid sequences. And once we get enough amino acids, check this out. Here is our mRNA sequence from codons in translating. Okay, so here we go. We're going to try and step 1, find the first letter in the codon in the inner circle. And I gotta find a place to move my head here. How about right there? There we go. So here's A. Check it out. There's A. All right. Right in the middle, the very, very innermost part of the circle here. The chart is, that's our first nucleotide. Okay. Then it says find the second letter of the codon in the second circle. Okay. Okay, so our second letter is G. Here's G. There it is. Okay, we're getting closer to figuring out what amino acid it is. And then finally, step 3, find the third letter of the codon in the outermost circle to determine the amino acid that the codon translates to. Okay, so our third letter is U. And guess what? There's U. So if we follow the amino acid sequence, we go A in the inner circle, G to the second circle, and U to the third circle. We find that it translates into SER, which stands for serine, and that's a type of amino acid. Okay, so our first one, I'm just going to write it out here in this sequence, is serine. Wow, this is really bad. Okay, you get the idea though. It's serine. Okay, that's our first amino acid in this sequence. Okay, and remember there are 20 different amino acids, so we have lots of different codons, but all those codons convert to an amino acid. All right, so use that chart here. See if you can't challenge yourself and figure out what the rest of the mRNA is. We have CCA, UGC, and GAA. Figure out what the other 3 amino acids are making up this sequence here, and the amino acids we can find on the very outside of the circle. Pause the video, see if you can't try it yourself. If not, I'm going to be pressing on. All right, so if you translated properly, you should have gotten serine, proline, cysteine, and glutamine. Okay, you don't need to know the specific names of the amino acids. The abbreviations should be just fine. Okay, but there's our 4 amino acids there, and this is how translation works. Okay, mRNA gets fed through that ribosome, and the ribosome uses its code in order to put the right amino acids in the right sequence. Okay, so all living things have the same code. mRNA is read one code at a time, just like you just did, through the ribosome until what we call a stop codon is reached, and this completes protein synthesis. So while you were looking on this this chart over here, you might have noticed some of these don't code for amino acids. They code for what we call, it just says stop over here. Okay, and how does a ribosome know when to stop translation? Well, it reaches one of those what we call stop codons, so like UAA, UAG, or UGA. Okay, so that's how translation stops, and that will complete protein synthesis. That's how we have a complete protein. Okay, so something that's going to lead us into our next couple units is how do we get new traits? How do we get new proteins from protein synthesis? Well, the short answer is that mutations occur. And a mutation is a random change in the sequence of nucleotides in an organism's DNA. Okay, so a mutation occurs to DNA. If you change the instructions, then you're going to get something, you're going to get a different product. If you change the system of how the cell is going to make that protein, you're going to get a different protein, right? So there's a couple different types of mutations that we're going to talk about. One is first called a point mutation, and that occurs when a nucleotide change causes one amino acid change. Okay, so take a look at this picture here. If we change CTT, and then all of a sudden, a mutation occurs, and what we get is CAT, we get a different mRNA sequence, and thus we also get a different amino acid as well. Instead of glutamine, we get valine here. Instead of glue, we get val over here. And that is what we call a point mutation, when one amino acid changes. Okay? But what if we take out a nucleotide, or what if we add a nucleotide? Okay, so this is our mRNA from before. This is the same one, AG, UCCA, UGC, GAA. Okay, what if we just took out this G? What would happen then? Well, remember, the ribosome reads 3 nucleotides at a time, right? So instead of these being AGUCCA, we're going to get a new, what we call a reading frame here. We're going to get new sets of 3. Okay, so it's going to go AUC, CAU, GCG, and AA whatever. Okay, so this is what we call a deletion mutation, where we get rid of one nucleotide, and it throws off the whole protein, right? So an insertion or a deletion shifts the sequence and results in what we call a whole new protein, and this is what we call a frame shift mutation, which can cause really big changes within an organism if the mutation is not fixed or if it's not addressed. Okay, so one little change in DNA can make a huge difference. Okay, so how is DNA like a language, and how is protein like a sentence? Well, think about it. DNA has the language of 4 different letters. We have a language of 26. Okay, and that DNA, that 4 letters, gets converted into 20 different amino acids. We use 26 letters to form words, and those words form sentences which have meaning, right? Proteins do a specific job. Okay? A protein doesn't, but needs instructions, needs language in order to get to that meaning, in order to get to that job, right? So that's kind of how DNA is a language and a protein is a sentence. We use that DNA in order to make a protein, just like we use language to create meaning and to create communication. Right? So then that second question was, how does a molecule like DNA determine your traits? Your combination of A's, C's, T's, and G's determines what kind of proteins your cells make, and what proteins your cells make determine your traits. Okay? And remember, your set of A's and G's and C's and T's are unique to anybody else that will ever exist or has ever existed. So congratulations. You're unique. You're special. And I hope you learned from this video. Please let me know if you have any questions, and we'll see you next time.